Well, good morning, everyone, and thank you for joining today's Garvan virtual public seminar dealing with how your genes may influence your individual response to COVID-19. For those of you who I've not had the pleasure of meeting yet, my name is Mara Jean Tilley, and I have the great privilege and pleasure of running the Garvan Research Foundation, the marketing and fundraising arm of the Garvan Institute of Medical Research. On our panel today, we have Priyanka Kalkani, who some of you may know from our public tour initiative back in the day when that were possible, and we certainly hope we can do that again soon. We've also got wonderful Joseph Hewitt as our moderator and IT help person. In terms of today's talent, we've got two of our truly wonderful scientists. We have Professor Stuart Tangye and Associate Professor Joseph Powell. And of course, last but not least, we have our big boss, Professor Chris Goodnow, Executive Director of the Garvan Institute of Medical Research. Before we kick off, I would like to take the opportunity and recognising that we are all dispersed at this time, but take the opportunity to acknowledge the Gadigal people of the Eora Nation, the traditional owners and custodians of the land on which the Garvan Institute is located, and pay my personal respects to elders past, present and emerging. And I think you'll all agree that on the eve of Reconciliation Week here in Australia, that acknowledgement has never been more poignant, not to mention what's happening in the States, which I'm sure everybody is seeing on the news at the moment. I would also like to acknowledge the individuals working on the front lines of healthcare, caring services, childcare, teachers. This has been a particularly traumatic time, I think, for everybody delivering those services. And we take our hats off to you, including the many Garvan clinician scientists who have been working in the COVID-19 wards and with their patients during this time. And of course, a special shout out to our friends and founders, St Vincent's Hospital, who've been doing an amazing job. Our hats off to you. So before we move on, a little bit of housekeeping. Now, a number of you, I think we've got some regulars in the room. Welcome to you. Great to see you again. I believe we also have some newcomers. So for those of you for whom this is your first time, the format for today will be, we'll have three presentations, one informal from Chris, two more formal presentations from Stuart and from Joseph before we move to a live Q&A. So for those of you unfamiliar with the Zoom function, if you look at the bottom of your screens, you will see there's a Q&A bubble, text bubble, to the bottom right of your screen. So throughout today's session, please do feel free to enter as many questions as you have as we go through the presentations, and I'll do my very best to get to those before we conclude today's session. Now, we are running a tight timeline. I've probably already spoken too much. So without further ado, let me introduce the Executive Director of the Garvan Institute for the official welcome. Chris Goodnow is undoubtedly not only one of the country's, but one of the world's most renowned and recognised immunologists. Chris joined Garvan in 2015 and is the proud recipient of the Bill and Patricia Ritchie Foundation Chair. Chris became Executive Director in 2018, which has just been absolutely wonderful for us. Chris is a rare entity in that he holds membership to not only the Australian and the UK, but also the American societies, which is a true honour and very, very well deserved. Chris has been taking a leadership position in terms of Garvan's rapid research response to COVID-19, and under Chris's leadership, our scientists have joined forces with their collaborators both locally and abroad to tackle COVID-19 from every angle, and particularly to leverage Garvan's existing and long-standing expertise in antibodies, immunology and genomics, and you're going to hear about some of that work today. But without further ado, I'm delighted to hand over to Chris. Chris, over to you.
Thank you, Mara Jean, and welcome everyone. This is the next best thing to all being in one place. Maybe it's the better thing. I know I've heard from some people at our last public lecture that we had people who had never been able to come into the Garvan for one of these, but because we're doing it by Zoom, they're able to come from places in the country, uh, and that's really wonderful. I think there are some silver linings to all this. So there's never been a more important time for medical research, and the achievements that Australia has shown in the last 3 months in flattening the curve really are based on all of the work that medical research that's been done in the last 100 years. The ability to sequence the virus's genome immediately after the outbreak started, to be able to develop very rapid testing to trace transmission in the community. These are all the fruits of long, hard medical research, some of it very basic and fundamental. So for example, the work that is happening here in the Garvan, sequencing the viral genome as part with our UNSW partners and the Southeast Area Health Service, that uses a technology that came from people just studying pores in bacteria, little holes in bacteria, with no particular thinking about how it might be medically relevant. And yet now that technology is right at the coalface of our battle with this virus. So medical research, very, very important, and medical research is the solution. Now at Garvan, when this outbreak happened, we realized we had a lot of really deep expertise across different disciplines that could be brought together to deal with this problem. And that's one of the things we're doing without losing sight of all the strength and expertise and ongoing work we're already doing in diabetes, cancer, bone diseases, immune diseases. But all that expertise does have a substantial part to play in COVID-19, and you're going to hear two really good examples of that today from two of our leading scientists, Stu Tangye and Joseph Powell. What I'd like to do is I'll briefly flag one thing that you heard about if you attended last time, which is how we're using gene technology to get an immune reaction, a human immune reaction, to happen in the laboratory better, faster, and more reproducibly than we could ever get a vaccine to produce therapeutic antibodies. And that's work that Daniel Christ's team at the Garvan has been leading. I got an update on that last night. They've made extraordinary progress. This is the most straightforward route to producing a treatment or a drug that would be highly potent and highly specific against the virus. They already have lead compounds, antibodies, about 11 of them, that are already extremely potent and specific. There's still a long way to go, but it's the fastest route we're aware of to a treatment or something that could give someone a month's protection. Now, the purpose of that line of research, it built on the fact that one arm of Garvan had a lot of expertise in therapeutic antibodies, and the fact that Daniel Christ had trained with the person in the UK that won the Nobel Prize 2 years ago for chemistry for inventing that way of bypassing the natural variability in our immune system to actually get an immune reaction to happen using gene technology in the laboratory. So we were really fortuitous that many of the people that have supported Garvan over many years had made it possible to have that kind of brilliant mind and brilliant team in place at the time that the outbreak started, and they could quickly expand their efforts on that one. Now, for the rest of the session today, we're going to talk about our own immune system, which is much more complicated, much more variable from one person to the other. And we're going to hear two really remarkable insights into that variability. So in a nutshell, the key thing to think about with COVID-19 is it illustrates that variability from one of us to the next in the way our immune system is wired up. Uh, it's not the virus that causes people to become very ill with COVID-19. It's actually the way our immune system reacts to the virus. And so, as you know, some people can have the virus and have no symptoms. They're shedding the virus, but their immune system tolerates the virus uh, and they go about okay. And then, of course, at the other extreme, 5 to 10% of people have a very extreme reaction to the virus. And it's that reaction that severely damages the lungs and ends up with people being in intensive care. Now, the real fundamental question is what's different from one person to another that we vary so much in the way our immune system reacts to the virus? And if we can get a hold of that information, that's the cornerstone for solving the whole COVID-19 crisis, because in the clues from that human variability, each of those are like experiments of nature that are telling us how to develop treatments to prevent that severe immune reaction without suppressing the whole immune response.
And so, Chris, if I or one of the lovely people in the audience were to have pre-existing autoimmune conditions, is it likely that we would have a more negative response to COVID-19?
We don't know yet, and that's one of the key pieces of research that will and answers that will come out of the work that Stu Tangye and Joseph Powell are doing at the Garvan. At the moment, I'd be speculating, and so that's the short answer. Let me tell you, just set the scene with very briefly. I'm conscious I've probably spoken too long, but just to remind everyone about our immune system. So our immune system is our natural defense force against viruses and other organisms. Every cell in our body actually is equipped with quite a few immune defense reactions at its disposal. And then, of course, we have millions of cells that are dedicated just to immune defense, to defending our body against viruses. Some of them fight hand-to-hand combat with infected cells. They're the killer T cells, for example. Some of them are the archers of our immune defense force. They sit in the trenches, but they fire off antibodies that home in on the virus wherever it is. Those are the B cells. And some of the specialized immune defenders of our body are kamikaze pilots. They basically blow themselves up wherever there's a virus and take out everything in the region, including the virus. And so it goes on. Enormous cellular complexity. But underpinning all of that and wiring up all those immune reactions in every cell in our body is thousands of genes. And those thousands of genes, remember we have over 20,000 genes in our genome, but the several thousand that are critical for controlling these immune reactions are the most variable genes in our genome. They're the ones that differ most from between myself and Mara Jean and everyone else on the webinar. And there are very good reasons why. And it's at the core of that genetic information that lie the answers to the variability of how each of us responds to COVID-19. And with that, that's probably sets the scene pretty well. The only thing I might end up with is a little bit of a hard sell that hopefully you'll be inspired by the things you hear today. And if you are, please think about giving to the Institute. Now more than ever, we are really in need of support, as you can imagine. We're still trying to do everything that we always care deeply about at Garvan and do COVID-19. And that will take as much support as the community is willing to support us with. So thank you, and I'll hand back to Mara Jean.
Thank you, Chris. Wonderful, wonderful and perfect segue. So everybody, our first speaker today is Professor Stuart Tangye. Stu is head of the Immunity and Inflammation Disease Research Theme at the Garvan Institute and the first Australian to very recently receive the President's Award from the Clinical Immunology Society of North America. He was also named a Clarivat Web of Science highly cited researcher. That may not mean much to the general public, but rest assured, it's a very important acknowledgement of Stuart's contributions to our understanding of the immune system. Stu is currently undertaking critical research to determine the genetic basis of severe COVID-19, and he's doing this in collaboration with a number of Sydney hospitals, as well as our friends at UNSW's Kirby Institute. They're currently analysing the DNA of both children and adults who've received a positive diagnosis for COVID-19 and ended up with the more severe symptoms that are being experienced for patients, despite not having had any other pre-existing health conditions. So that's obviously quite interesting. And we're hoping that this research may point to new therapeutic targets for the disease or could uncover existing medication that could be repurposed to help protect and treat people having the worst response to COVID-19. Stu, over to you.
Thanks very much, Mara-Jean. Hopefully you can hear and see my screen from my home. So welcome everybody. Thanks a lot for joining us for the Garvan public seminars. These are a great opportunity to bring a little bit of Garvan to the public, and in this case, literally bringing us into your room. So thank you very much for the time taken to tune in this morning and hear about the research that Garvan's doing in the space of COVID-19, but obviously today's focus is really on understanding the immune system. So I'll follow up a little bit of what Chris started telling us about in terms of the immune system in general. And of course, as Chris said, the immune system really is our defense mechanism against infection. And we know that very well because when our immune system works, we're generally healthy. We've seen great advances in the preventing of infectious diseases by vaccination, for example. So the immune system is really important for stopping us from getting really sick from infectious diseases. There's also important roles for the immune system in detecting and eradicating some forms of cancer. So your immune system exists all over your body. It is comprised of many different cell types, and these are all present in your bloodstream, but also in many of your bodily organs. So you have white blood cells, which are these lovely looking large white blood cells, lymphocytes, macrophages, some of the cells Chris was talking about, T cells, B cells. And these cells really communicate and work together to stop us getting sick from whether it be a bacterial infection or a parasitic infection or fungal or mold infection, or as we are talking about today, viruses. So being of the age that I am, I like to relate things back to something that I'm quite fond of, which is, of course, Star Wars. So to us, a simple way of looking at it is, this is when you get infected, there's this huge proliferation or expansion of invaders, in this case, the stormtroopers being pathogens, but there's the good guys, which of course is the immune system. And when these two forces collide, epic battle ensues, and hopefully at the end of the day, the invaders are destroyed and wiped out. And we go on to be healthy. And our immune system is often so good at this that when the pathogens come back for a second time, we're really primed and ready to go. And we don't even know we get reinfected following exposure to the same bacteria or virus at a later time point. But sometimes the force, being our immune system, is just not strong enough, or it behaves in the wrong way. And in that case, our immune system can actually end up doing some damage to ourselves rather than good. So how does this operate? Why can that be so extreme? Why do some people deal with infections very well? Why do other people develop really severe consequences of viral infection? Why do other people develop really severe infection due to the virus? And the answer more than likely lies in our genes. And our genes are really what make us who we are and how we behave. So I'm sure you're all familiar. Genes are the building blocks of our life. They're present in our DNA, and our DNA is packed on all of our chromosomes. And within every cell, we have 23 pairs of chromosomes. They live inside the nucleus of our cell, and this is what we end up with at the end. And as Chris mentioned, the number of genes in our body is known. There's between 20 and 25,000 genes in humans. But it's remarkable to think that there's also very much genetic complexity across different species. So believe it or not, there's actually more genes in rice than there are in humans. And there's actually more genes in water fleas than there are in humans as well. But within humans, there's about 20 to 25,000 genes, and about 2,000 of these genes are really important for the immune system. That's been calculated based on many studies. And we know from studies over the last 30 years or more that we've identified nearly 450 genes which can cause severe immune diseases. These are mostly infectious diseases, but they can also contribute to allergy or autoimmunity or serious inflammatory disorders like inflammatory bowel disease, for example. So it's the nature of our genes and the composition of our genetic makeup which really dictates how we respond at the cellular level to infection. And I want to illustrate this very simply by talking about two boys. This strapping young lad was born in 1970. He didn't quite grow up to be the big wave surfer that he wanted to, but he's relatively content as an immunologist. He grew up to have a very normal childhood, you know, playing sport, rolling around in the dirt, getting hugs from his mother, and growing up as you do. About a year later, this young boy was born. His name's David Vetter, and you may know him better colloquially as the boy in the bubble. He spent 12 and a half years of his 13 years in a bubble. And that's because he was incredibly susceptible to infection. He couldn't play sport. He couldn't go outside. He couldn't, be kissed and so on by his family, not unlike what we're experiencing now. And that's because his immune system was so vulnerable that he was susceptible to infection with almost everything. So the simple difference between these two boys who were born at a very similar time in life and grew up under very similar circumstances in terms of the era is that David here had a single genetic defect, and that caused a disease called X-linked severe combined immunodeficiency. So in David's case, he had this single gene that didn't work. And so instead of having normal DNA, he had an error or a mutation here. And that actually prevented the development or function of one of the major subsets of his immune cells. And as a result, he was incredibly susceptible to infection with all of those pathogens that I showed you before. Now, this is an extreme example, and these are rare conditions. And sometimes you can have these holes in your immune system, which make you susceptible only to a particular type of virus. So I've done a lot of work on a virus called Epstein-Barr virus, which causes glandular fever and is relatively harmless in most of the population, but in some cases can be fatal if your immune system doesn't work properly. You might just be susceptible to some fungal infections or some types of bacteria. But in this case, this was an extreme example of impaired immunity, which really meant he had to live life in a sterile bubble. Now, we don't do that anymore, of course. We have ways of treating this disease, but that is a clear example of one gene making a huge difference to your immune system. So what are we doing about this? So Garvan is part of a global initiative, which is called the COVID Human Genetic Effort. It was established by these two colleagues and friends of mine, Jean-Laurent Casanova, who's in Rockefeller University in New York City, and Helen Su, who's at the National Institutes of Health in Bethesda near DC. And what Jean-Laurent and Helen Su have done is corralled many of their colleagues and investigators literally around the world, and I'll show you that in a second. And what we're trying to do is deploy all of our resources as much as possible to recruit people who have had severe COVID-19 and look at their DNA and try to identify what differences they may have, which have really impacted the way their immune system works so that they develop really severe disease under conditions which they're otherwise healthy. We're also looking for people who have been exposed to exposed to SARS-CoV-2 and haven't really developed disease under situations which you think they may have. So we're really trying to understand and identify how different genes influence the immune system and how this then controls the way each of us is going to respond when we get exposed to this particular virus. So this goes back to something Chris mentioned as well. This sort of shows what we're trying to do. This is the general population, and we've never seen SARS-CoV-2 before. So our immune system is naive to this virus. We see lots of viruses and bacteria all the time, but this is literally a new visitor to our world, and we don't really know how to deal with it. We've seen similar viruses in the past, but this one in particular, we've never encountered before. So it's going to have a very different effect on different people. So we already know that the elderly population are more susceptible to developing severe disease. And we also know people who have what's called comorbidities or pre-existing health conditions like diabetes, heart disease, lung disease. They're also more vulnerable to infection. So some of those outcomes can be pretty well guessed based on pre-existing health conditions. We also know that the young are relatively protected from developing severe disease. However, there are rare individuals who are otherwise healthy who are developing extreme COVID-19 disease. And there's also people in, for example, the older population who may be being exposed to a lot of infected individuals who actually remain quite healthy. So what we're trying to do, we're trying to identify these people, the red individuals and these mustard colored individuals within the population of virus positive individuals who have very different outcomes, very sick under unexpected conditions, and then very well under unexpected conditions, and look at their DNA to try to see what's going on. So I said, this really is a global effort. Here's Garvan sitting down here. We are the Oceania hub for DNA sequencing. That's, of course, being done at the Kinghorn Center for Clinical Genomics. And we are part of this global initiative, which clearly stretches across all corners of the globe. And it's a really collegial effort, which works together and meets weekly, to discuss the outcomes. Colleagues, we've already started generating a list of genes, which we think are likely to be responsible for this different types of outcomes, but that needs a lot more validation, obviously. We here have now rolled this out across Australia, and we're working with colleagues locally, very locally, St. Vincent's Hospital, for example, but also many hospitals across New South Wales, and also hospitals in most states and territories across in WA, north to us in Queensland, Canberra, South Australia, and so on. We've also got colleagues at Starship Hospital in Auckland who are also helping us out, particularly identifying patients who may not be able to access the best healthcare systems, for example, some of our Pacific neighbours who need help from ourselves or colleagues in New Zealand. So it's a global effort. It's being led very strongly here from the Garvan for the Australian perspective. We are very thankful for the UNSW for providing us with initial funding to get this going, and the many people at these different sites who are really underpinning the recruitment of patients, the testing of patients, and then of course people on site at Garvan who are really doing the hard yards of the DNA sequencing and the analysis to try to find genes which are going to tell us, why do people get so sick? What parts of the immune system are really important for protection against this disease? Can that inform new therapies? Can that help develop vaccines? And can we find ways in which we could prepare people to resist this disease into the future for people who have not yet been exposed to the virus? So I'm just going to stop there and take questions now. We're at the end, but I'll pass back to Mara Jean. And thanks again, everyone, for tuning in and listening and being part of the virtual global family. Thanks very much.
Thanks, Stu. Fantastic presentation. Now, I do have a question for you, but before we get to that, I'm really stuck on this issue of genetic similarity to rice, water fleas, and chickens, with just, from what I could see, 4,000 differences in protein coding genes from a chicken to a human. Can you just extrapolate on that point a little bit for everyone's comfort, please?
Well, that's probably a little bit of a simplified picture, but it is remarkable. Between humans and our nearest primates, the great apes and so on, there's very little difference. There's like 1% difference in our genes, 97 to 99% similarity in our genes. And then, of course, all of us, at least if we think of vertebrates, chickens, we all have to do the same thing. We all have hearts, brains, some bigger than others, blood systems, immune defense systems. So the fundamental building blocks are pretty much present in all of those species, probably not rice and water fleas. But we need those organ systems to function and to survive. So those building blocks are similar across the board. Of course, there are differences, and there's genes that are present in some species, but not others. But for fundamental survival, you need a common system, and that's pretty much part of the shared building blocks of life.
Okay, thanks, Stu. Perhaps we should have a dedicated session in future to unpicking this. Really fantastic presentation, Stu. So from the cases that you've seen and sequenced thus far, are you seeing any indication of this idea of potential herd immunity or potential individual patient immunity? And is there potentially a genetic reason for people developing that or not? Or is it too soon to say?
It's pretty early to say, Mara-Jean. For herd immunity, the way herd immunity works is you need exposure. And the way you get exposure is either through natural infection and natural exposure of the virus, or vaccination. And for this sort of virus, the calculations are we need about 60 to 70% of the population exposed to have some type of herd immunity in the community. Now, although we've had 7,000 cases and over 100 deaths, and Australia's done very, very well with this. If you work that out, our level of exposure is very, very low. It's less than 10% of the population. It's way less than 10% of the population. So we're nowhere near the minimum threshold that is predicted to be needed for herd immunity for this virus. That could change once we get vaccinated, of course. And you'll always hear about measles outbreaks in the context of people stopping getting vaccinated. And that's what lowers the coverage of the vaccine. And then that's when herd immunity sort of drops off a little bit. So we can't get to herd immunity until you have much greater levels of exposure, either naturally or by vaccination.
Fantastic, thanks, Stu. Okay, we're going to move on to our next speaker, Associate Professor Joseph Powell. Joseph is the director of the Garvan Weizmann Centre for Cellular Genomics. Some of our audience members here today may be aware that Garvan has a strategic partnership with the Weizmann Institute in Israel, and we have a huge joint effort in single cell sequencing. Joseph is also the head of the Single Cell and Computational Genomics Lab at the Garvan Institute, and his combined expertise now sees the centre owning the rightful position as the third largest throughput of single cell data globally, alongside our friends at the Broad Institute and the Sanger Institute. So Joseph's done a remarkable job really building up the capacity and the outputs of the Garvan Weizmann Centre. Joseph and his team are currently involved in leading a global effort to uncover how the genetics of different immune cells determine susceptibility, severity and outcomes for people with COVID-19. The team is leveraging existing genomic data available through one of Joseph's long-standing research initiatives, a program called 1K. 1K. This is a pioneering study investigating the genetic data of single cells from more than 1,000 healthy individuals who are participating in the study. And basically the end aim is to discover how genetics can control and influence the immune system. So Joseph, welcome, over to you.
All right, good morning, everybody. I'm just going to share my screen for a moment. Is that coming through? Perfect. So, so good morning. I'd like to apologize initially for my rather ungroomed appearance. I think I was preparing for a slightly more apocalyptic scenario than we've had. So I'm obviously extremely happy that's not the case, and I should probably visit a barber pretty soon. So I'm going to talk today about our work around the genetic control of severity or severe reactions to COVID infections. And, you know, as we all know, this is one of the really, you know, the most scary, but also one of the most fascinating aspects of this virus, in that people that are infected with the virus, many can essentially have no symptoms. They can be asymptomatic. We don't really know how many people have that at the moment for obvious reasons, but it's certainly quite a lot of people. And then you have people that develop symptoms And we have a huge range of different symptoms that they can have. A lot of people have really mild conditions, you know, a dry cough, through to obviously the really scary severe symptoms, and of course the critical conditions where people are on ventilators and life support. So this, you know, from a scientific perspective, to me, you know, is actually really quite fascinating. And we have this question that's posed when you look at this, this result of why do we see such a variable response to infection with the COVID-19 virus? So as Chris and Stuart have just really beautifully introduced and then outlined, the thing that's quite apparent is that certainly for the patients that have a really severe response, it's not so much the virus causing that, but really, you know, let's call it the overreaction of our immune system, which causes these particular symptoms. Now, given that the immune system is controlled by genes, and we have different copies of different genes in different individuals, it really focuses our question about why do our genes play a role in contributing to the severity of response. So to me, the question really is not so much how does COVID impact on our genes, but how do our genes impact on our response to COVID-19 infection. So I'll just, you know, duplicate a little bit of the discussion from Stuart here and just go through my take of some of the background here. A gene is obviously, you know, a segment of DNA that encodes for a particular type of protein. Each gene has a different type of protein that it encodes for. And we all carry different copies or different versions of these genes between us. And it's these different copies that really make us different from one another, both in our appearance and also our susceptibility to diseases. And genes control, you know, pretty much every process and system in the body, including how the immune system works and how it, of course, responds to infections such as infection with the COVID-19 virus. Now, the other really critical piece of information for me is that we also have differences in the switches that control how genes are turned on or turned off. And so these differences in the switches between different people also have an effect on things like our response of our immune system and susceptibility to different diseases. So I'm going to go through in a moment and talk about some of the work that we've been doing and bringing in a lot of the cellular genetics into understanding our response to COVID infection. But I wanted just to pause for a moment, particularly in light of Stuart's presentation, to make, you know, what I feel is kind of an important distinction about the way that our genes contribute to things like diseases and how our genes contribute to the way that we respond to infections like COVID-19. So there's essentially two kind of scenarios that we have. One scenario is that, which is often called by geneticists called rare or sometimes monogenic conditions, where our probability of getting a disease or our probability of having a severe reaction to COVID-19 is really controlled by one gene, and then it's really the luck of the draw of whether you've got a good copy or a bad copy of that gene. So using the analogy of dice, you know, really we have one die, and if you're fortunate to roll a 1 to 5, you have a good copy of that gene and you have potentially an asymptomatic or a very mild response. And if you're unlucky and you've inherited a bad copy in a roll of 6, you have a very severe reaction. So this is one scenario. The other scenario is situations which in genetics, again, we call common or sometimes complex conditions, where really we have a very large number of different genes, each of which have some role in contributing to our severity of response. So to follow through on that analogy of dice, it's not really like rolling one dice and hoping you don't roll a 6. We have to roll lots of dice and really sum up the different effects across all the different dice. And really then it's the combination of your copies of either good or bad genes that denotes how mild or how severe your reaction to a COVID-19 infection. And so Stuart's work really focuses on this rare set of conditions, and the work that we've been doing is focusing on these common conditions. Now, the reason why I've sort of labored this point a little bit is that the way that we study these things in both the experimental design and also the way that we use the results to inform therapies is a little bit different. So in the complex set of scenarios, or the common and complex set of scenarios, one of the really key important factors is that, one, we have literally hundreds of genes that can contribute to a different response, and every individual will have a different permutation of those genes. But the other important thing is that the genetic effects largely act on changing the switches that control the genes. So really the regulation of those genes. Now, the kicker and the bit that makes it really complex is that the way that these control mechanisms work differs between different cell types. So you have the same genes in every cell, but the way that they work and the way that those switches work is different in different cell types. So for us, this creates, both, a sort of an exciting opportunity, but also a challenge, because as the immune system is comprised of a really wide variety of cell types. They have very distinct functions. They all carry the same genes, but the way those switches work can be quite different. So how do we solve the problem of bringing what we know about the genetics into how those genetics work in individual cells? How do we really solve this problem? And for us, we really need two pieces of information to understand how our genes control the severity of COVID infection. First piece of information we need is literally which of the 25,000 genes are associated with severity. Which genes have good and bad copies, which denote whether you will have a mild or severe reaction. And secondly, we need to have knowledge about how those genes are controlled in each of the different immune cells. And we've been really fortunate from a scientific perspective as this pandemic emerged and obviously rapidly evolved, in that the scientific community very quickly came together to generate an international consortium to understand the genetic effects, identify those genes. This is a very similar initiative to the one that Stuart just mentioned, but instead of focusing on the rare types of genes, it's focusing on these common types. And the second was a study which Mara-Jean introduced called 1K1K, which was a large program of research within the Garvan Weissman Center that was already underway of trying to understand how these genes are controlled and switched on and switched off in each individual immune cell. So I'll just briefly introduce these two programs of research. So the Host Genetics Consortium, as it's called, is an international program involving, I think now about 150 institutes. We are involved, as are a number of other Australian universities and institutes. Unfortunately, I couldn't get the map to go large enough to show Australia, but rest assured we are there. And what this is really an initiative to compare the genome sequences of people that had a very severe reaction to the genome sequences of people that had a very mild reaction, to start identifying which positions, which genes are associated with the severity of response to COVID-19 infection. And the second piece of information in this 1K cohort, and I won't go through all the details of this because of time constraints, but this was a very large initiative which we've established to map how the genetic differences between individuals control genes in each of the different immune cells. And the reason why we were working on this was that obviously genetic effects on immune cells have effects in hundreds of different diseases, autoimmune diseases, the way we respond to cancers and immunotherapy and many other things. And so we are using this resource and this research cohort to understand these effects on diseases. But as the COVID pandemic emerged, it gave us this opportunity to bring together the genetics with the cellular genomics to to be able to understand how genetic effects work at the level of an individual cell. So if we're able to solve this problem, if we're able to find which genes control severity of response, and we're able to understand which immune cell types they are working in or not working in, it gives us this target for developing, potentially developing therapies that are able to hone in on individual genes in individual immune cell types. And also potentially it gives us this ability to be able to predict for an individual in the population how severe their infection or their reaction to an infection would be before they are exposed to COVID. And we can do that by using the genome sequence information. So what we do is we bring together these two major research cohorts. We apply some relatively fancy statistical or machine learning techniques to identify these targets of how genes work in individual cell types. So just to give you a complicated figure of what we get when we do this, this figure here, each row of a figure denotes a different type of the immune cells. And each column, and there's a lot of them, denotes a different gene that has been shown to be associated with the severity of COVID-19 response. So the first take-home message is that there's a lot of genes. This is a very complex scenario, and you can see that the way that that gene works or acts in each different immune cell is quite variable. So this gives us that map of being able to get down to resolving how the genetic effects work in different immune cells. So just to sort of highlight one particular example, what we can then drill into with this is seeing how the different genes that different individuals carry work in individual cell types. So each one of these little figures on the right-hand side here is a different type of cell, immune cell, and it's showing how different copies of that gene work in or switched on or switched off in that particular type of cell. So there's basically 3 different versions of this gene in this population. So using this information, this gives us this resolution of being able to get down to how the cell types, different immune cell types contribute. And it means that we can use the information to start determining how an individual's genome sequence denotes their particular risk of having a very severe reaction. So I'll finish up there, and I'll just summarize, what I've been talking through. And the first is obviously, I think it's hopefully quite apparent that our body's response to this infection is to a large extent due to our genetics, or at least that's where the evidence is pointing at the moment. And there are a lot of genes that we all carry, and we all carry different copies of them that sum up to influence how severe a reaction will be. And we think that most of these genes seem to be functioning in a really limited number or type of immune cell. And so we can use the type of studies that we're working on here at Garvan and across with our international collaborators to start pinpointing the function and what's really happening in the different immune cells to potentially start developing therapies for this. So I'd like to just finish up there with some acknowledgements. Thank you all for your attention. I would like to particularly point out some work from the Garvan Weissman Research Center of Sarah Balouz, Jose Alcacera and Seyhan Yazar, who have done much of the actual work on this, and of course acknowledge the fantastic support we've had both from philanthropy and of course from the state government and competitive grants. Thank you very much.
Brilliant. Thank you so much, Joseph. Now, before we turn to the live Q&A, Joseph, a question from me. Is it true that research is showing that men have a particularly worse response to COVID-19? And if so, why?
Yes, that's true. It does appear that is the case. I think the short answer is we don't know why exactly. There are some potential genetic reasons why that will be around the way that our X and Y chromosomes control the switching on and switching off of different genes. And that is something that we've started to look into with this research program. But I think there's probably a very wide range of reasons for that particular observation, both genetic and potentially non-genetic.
Thanks for that, Joseph. Is there any possibility that the wives' tale around man flu could actually be legitimate?
Quite possibly, but I think when you get to that severe end of the spectrum, it's hard to be faking that set of scenarios.
Okay, well, perhaps we'll revisit that topic at another time. So we've got some good questions and comments coming through on the Q&A function. So I'm going to kick off with the first one. Chris, I'll give this one to you, please. How do we know or define that COVID-19 is a virus and not bacteria or a parasite?
Okay, that's a good question. The fundamental difference is that bacteria or parasites, well, parasites you can see them, like roundworms or ticks. Bacteria we can't see, but they are free living. They're their own organism, and they have everything they need, more or less. Whereas viruses are, in a peculiar way, an extension of our own body. On their own, they're not alive. They're only essentially a package of DNA or RNA. In the case of SARS-CoV-2, it's RNA. Ribonucleic acid is their genome, about 30,000 base pairs of ribonucleic acid, and that's really all there is to the virus, and some proteins that package it up and switch that RNA on once it enters our cells. But most of the machinery that the virus needs to replicate has to be co-opted and stolen from our own cells to make more copies of the viral RNA and the viral capsid and the spike protein. So how do we know that the disease is caused by a virus? The key thing is there are various ways of, the most important thing is you can show that there's something in people's nose which, if you run it through a filter that would take out bacteria, there's still left very tiny particles that when you put onto human cells in culture, trigger those cells to blow up. And what we call plaques, virus plaques. And from that, we can actually count how many virus particles were in a sample. For a long time in science, that was the only way that we could detect a virus, going right back to the great Macfarlane Burnet, who sort of invented some of those plaque virus counting methods for influenza and measles. But of course, nowadays we do it by polymerase chain reaction, PCR, which essentially allows us to, that's what's happening when we go, as I did on Monday afternoon. I went up to Northern Beaches Hospital because I had a bit of a runny nose, and they swabbed the back of my nose and throat. And then that went off to a laboratory, that swab, and they essentially did a polymerase chain reaction to amplify any viral RNA from the SARS-CoV-2 virus thousands of times within the space of a couple of hours. And if there had been even a few copies of the virus, it would have yielded a positive. In my case, it was negative. I got a nice phone call yesterday. I can come out of isolation now. But that's how we know that it's a virus. And that's really has been a huge game changer relative to even the SARS outbreak in 2003, where it took many months before we had a hold of the virus and had the sequence of its RNA to set up one of these polymerase chain reaction tests.
Fantastic. Thanks, Chris. Joseph, I'm going to give you the next question. Following recent research, is it correct to say COVID-19 is more a vascular disease rather than a respiratory disease?
Yeah, that's a really good question. So the particular receptors on cells that allow infection of the virus expressed in certain points in the respiratory tract, and then at many other points in the human body as well. And there's been a lot of very nice work to show that. But I think what is known about let's call it the severity of the symptoms, and we have a set of symptoms that involve, inflammation and damage to the vasculatory system. That's one of those set of scenarios where we would anticipate that a good fraction of that is really being caused by the immune system, or the immune system's interaction with other cells causing that particular set of conditions. And so this is why when you have some individuals that have, one set of symptoms really severe, maybe, inflammation in the vasculatory system, whereas other individuals have a, very localized to the respiratory system. One of the potential reasons for that would be the different permutations or the different rolls of a dice of the different genes that they carry, and then the way that the immune systems will work at causing a severe effect in one system and a severe effect in the other system.
Chris, you'd like to add something?
Oh yeah, I just wanted to add on because I think that's such a fascinating question. I think you're probably aware that one of the best laboratory predictors for someone progressing to very severe disease or ICU is evidence that the blood clotting system is being fired off. In particular, a product of clotting within the blood vessels called D-dimer, which has been shown over and over again in many studies now to be probably the best bad predictor. One of the questions is, well, what's triggering the clotting? And one of the things that's very interesting, I mentioned at the beginning that every cell in our body is equipped with its own defense response. One of those defense responses involves a gene that my team, together with a group at Genentech in California, discovered 5 years ago called Gas-dermin D. It turns out to be a way that cells in our body fight infection by essentially blowing themselves up with little holes in the membrane of the cell. And one of the things that comes out of that hole is a thing that triggers the clotting cascade. And so there's a very interesting possibility that what's triggering the clotting is essentially an unbridled immune response by a whole range of cells within our lungs to the virus or to the inflammation the virus has set off. So the end result is clotting in the vessels, similar to what happens if you've got sepsis. But that's essentially a downstream part of a manifestation of many cells in our body over-responding and essentially blowing themselves up.
Thank you, Chris. Now, I am conscious of the time, and I'm going to have to whiz through these questions and perhaps double up with any duplicates, etc. For anybody who does need to sign off right on 11 a.m., apologies that we will lose you. There will be a recording of the session available on our website after, hopefully within a day or two's time, if you'd like to come back to see the questions. The next one, Stu, I'm going to give to you, please. Just want to know your thoughts about susceptibility of COVID-19 amongst Indigenous and in particular Black communities around the world. This has been heavily reported around the world, and I wonder if it may have something to do with genes as opposed to ecosystem socioeconomic status in general. Hashtag Black Lives Matter.
That's absolutely a great question. And we have seen the influence of the incidence of disease across the board being greatly affected by ethnicities, which obviously comes back to genetics. But there are compounding factors. Clearly, with our Indigenous population, they've suffered great health due to genetics, but also clearly due to socioeconomic situations. So epidemiologically, we are trying to work out what the incidence of infected people are with severe disease, whether they are, for example, African American, Caucasian, and so on. And this is being done particularly in the Northern Hemisphere, where the cases are much, much more voluminous. So we can get a very clear distribution of the ethnicities which predominate the severe cases. That's not something I'm doing per se, because I'm not an epidemiologist, but that is something that's being discussed in these forums that I'm part of. From our perspective, I think it's very important that we keep an eye on the Indigenous population. There are cultural challenges in working with Indigenous Australians. It doesn't mean it's impossible, but quite appropriately, there are cultural sensitivities in terms of doing DNA research on Indigenous Australians due to particular cultural beliefs, which I completely respect. But I think it's important that we recognise that there possibly could be greater adverse impact of viral infection and many infectious diseases in our Indigenous community, and we can try to look into that. But we need to address that more front on.
Thanks, Stu. And if I understand correctly, we've thus far managed to prevent the spread of COVID-19 into those most vulnerable communities and populations, which is certainly an achievement. The next one, Chris, is for you. Is there any indication at this stage which of the many immune cell types may be of the greatest importance in having critical genes which may predispose to severe type responses to SARS-CoV-2 infection? And this is from our friend Anne.
Hi, Anne. You know, the news is changing on a monthly basis. The deepest evidence actually comes from SARS in 2003, which points to a range of cell types. But, you know, at one level, there's a suggestion that, well, what was clear in SARS is people that made antibodies strongly in the first 2 weeks of infection, that was correlated with having a worse disease, paradoxically. But correlation doesn't necessarily mean it's the antibodies that were triggering it. It could have been that the other arms of the immune response were firing off more strongly, and that led to the higher antibodies. What's very clear is the macrophages, the cells that gobble up all sorts of damaged cells and particles in the lungs. They're probably very important, and that was certainly implied both in SARS and is coming out in SARS-CoV-2 with our partnership with the Weizmann Institute. I might let Joseph speak a bit to that, and then of course the other place where we'll get answers to that's where genetics comes in. Is the trouble with correlation is that you don't know if it's cause or effect. And so one of the things that I'm really inspired about what both Joseph's doing with common gene variants and what Stu's doing with rare gene variants is that from that will come the answers to your question, Anne.
Chris, that's actually a perfect segue to our next question, which is to do, the question specifically is how long has this global collaboration research been running? From the time point, I'm not 100% sure if it's Stu's or Joseph, so why don't we get a response from both of you, please? Stu, do you want to start?
Yeah, sure. So for the human genetic effort that I was referring to, which is the monogenic, looking at single gene variants, I'm on the steering committee of that consortium, and we started assembling the sites in early March, and we had our first meeting at the end of March. So it's been going almost for 3 months. It took a few weeks, obviously, to sort things out, and it's still taking a little bit of time for various centers to get things like ethics approval. But we are now in that position, and we have St. Vincent's right next door, who has 300 patients who they can recruit, who we can work with. But essentially, it's been about 3 months, Mara-Jean, 2 months in full swing, a month to get things started.
Thanks, Stu. Joseph, do you want to talk a little bit about the Weizmann Institute, but also your broader collaborations?
Yeah, so we've had a long running collaboration both with the Weizmann and other institutes to look at that cellular control of different genes. But the particular international consortium that was put together to look at identifying which genes control a severity of response started at a similar time to the one that Stuart is co-leading, so early March. And, you know, just as a general comment, I mean, from my perspective, and this has been particularly nice with working from home, I mean, it's been absolutely phenomenal to see the collegiate collaboration between, like hundreds of researchers globally to solve some of the challenges here. And, I think it's been, a really inspirational part of what's happened in this scary time. So it's been very rapid. These collaborations and these types of consortiums in genetics are common, but they do not normally happen this fast or with this much ease of getting this many people together.
Thank you, Joseph. That's fantastic. Next question. Chris, I'll give this one to you, please. The problem for most or almost, the problems for most or almost deadly disease is our immunity, which is the fundamental block for cure and recovery. What strategy will be put in place if the substance used to stimulate the production of the antibodies, I assume this is Prof Christ et al., that would provide immunity against one or several diseases such as COVID-19 and other viruses? Sorry, does that make sense?
Yeah, I think so. I think I know what you're asking. There are two parts to that question. One is, for the antibodies that Daniel Christ's team is developing using gene technology in the laboratory, one of the elegant things he's able to do is to they've expressed the spike protein not only from SARS-CoV-2 virus, our current enemy, but SARS and also the bat precursor of the current virus and the virus that was related to it that came from pangolins. And so what the team has already got is they're finding that many of the antibodies only bind one virus and not the others. But they are finding some antibodies that bind 2, 3, or 2% of the antibodies bind all 4. And so that gives us a pathway to possibly produce an antibody drug that would fight a wide spectrum, both of this current virus and others to come in the future.
Great, thank you, Chris. We have somebody in the audience who's curious to know, are any of you geneticists?
Just a little bit.
The answer is yes. I think it's fair to say for all 4. There have actually been a couple of questions coming in about epigenetic factors. What about epigenetic factors affecting those key immune genes?
Well, that's a Joseph question.
Yeah, this is, look, without a doubt, epigenetics is probably one of the major contributors to the way that those switches work in different genes and different cell types. One of the really fascinating things about epigenetics, so these are the sort of the modifications of our DNA and indeed RNA, is that they can be, the variation in the epigenetics can be partly due to our underlying DNA sequence data. So it's a genetic effect on epigenetics, if that makes sense. And then the way that different environmental and different exposures work. So there's I'd say a large amount of evidence that it's almost the interaction of those two pieces of the jigsaw that affects that kind of variation. And so this is the kind of the final piece when we talk about starting to understand some of the genetic components of mechanisms is working out how the epigenetics affects the switching on and switching off of different genes.
Thanks, Joseph. That's great. Now, I do apologise, everyone, but I think we've got a hard stop coming up. We've got a number of questions around susceptibility to COVID-19, and I think, Stu, you might be the best person to address that.
Well, thanks, Mara-Jean. So there's one question about common variable immunodeficiency. So this is a condition which renders people immunodeficient. It's not really known what causes it in terms of genetics. So part of something that I'm very interested in is the impact of infection on people who are immunodeficient or immunosuppressed. We were quite concerned that individuals who are immunodeficient, either genetically or for some other reason, or immunosuppressed, for example, because they have autoimmune disease and are on immunosuppressive drugs, or cancer patients who are on chemotherapy, may be more vulnerable. So we actually did a survey across the world looking for rare patients who have known genetic defects that cause disease and become virus positive. So we found nearly 60 patients, which doesn't sound a lot, but in a short period of time for rare diseases, that was pretty good. And pleasingly, to our pleasant surprise, the severity of disease in immunodeficient patients was not too much more severe than what we see in the general population. The same comorbidities existed. The age of susceptibility was lower, generally speaking, than sort of the elderly population. But it almost seemed that to be a little bit immunosuppressed wasn't a bad thing. And that's possibly because we do see this hyperinflammatory response, which is causing severe disease. So it's not actually the virus that's causing the severe disease, it's the immune response to the virus that's causing a lot of the problems. So being a little bit immunosuppressed may be advantageous. Now, that's not published or anything, that's my interpretation of the data. But we're looking to sift through that data in more detail very soon. And that's also seen, there's been some studies in people who have had organ transplants. So if you have an organ transplant, you generally are on immune suppression for quite a long time. So in Italy and Switzerland, which have been hit very hard by SARS-CoV-2, there were, again, concerns there, but the organ transplant patients they identified who were on immunosuppression, who were virus positive, they didn't have severe disease. So, you know, it's a slight silver lining despite all of the other indications that are going on at the moment.
Thanks for that, Stu. That's fantastic. And apologies, everyone. We're not going to be able to get to all of your questions, but please do send them through to the Garvan Research Foundation via email if you're really keen to get something addressed offline. Thank you, each and every one of you, for joining us today. Our donors, our partners for the future, our board directors, our volunteers, our advocates. We are absolutely privileged to have your support, particularly during this time. We do thank you. As you know, we're still learning how to conduct our virtual seminars with greatest efficacy. So please do send through your feedback. All feedback, whether it be positive or constructive, is very warmly welcomed. We have a number of other virtual public seminars coming up, so please do check out the website. Thank you. We hope you're all staying well, staying safe, staying warm and staying sane. And thank you for joining us today. We really value your time and your attention. All the best.
Thank you very much.
Thanks, everyone.
Have a good day, everybody. Thanks.